conjugate, and therefore it reduces cytochrome C and apoptosis initiating factor release, and therefore protect the cells from apoptosis. And this pathway would also provide metabolites and energy for DNA damage repair to generate the metabolic factors for detoxification and inactivation of chemotherapy agents. And it increases the breakdown of the extracellular matrix, facilitating the cell motility and invasion through lactic acid and secretion of the acid into the matrix. So glucose would also help the cells to evade the assault of chemotherapy. A lot of the drugs, called xenobiotics, and the xenobiotics are metabolized by the cytochrome P450 pathway. These enzymes use the NADPH to metabolize the drugs into polar metabolites, which are then deactivated or further conjugated with glucuronide in the kidney and for excretion, elimination in either the kidney or in the bile. Now, in this process, for the NADPH, it is generated by the glycolytic pathway, as well as the metabolism of pyruvate. And the NADPH is generated through the pentose 6-phosphate pathway, coming this way. And the NADH is needed for the glycolytic pathway, and the NADH is also regenerated when pyruvate is converted to lactate. And the NAD is very important, is needed for the ATP ribosylase transfer to the DNA when the DNA is damaged by this enzyme called PARP. So, as I pointed out earlier, the carcinogenesis process involves a series of genetic changes. Right? And these genetic changes are not random. And typically, a series of genetic mutations, translocation, deletion, or amplification, or epigenetic changes, for example, through hypermethylation of promoter regions, etc., changing the expression of genes. And this happened to usually a key set of genes that initiate the process. And from these genes, they require the normal cells. These are required for normal cells to be transformed into malignant cells. And the Wellcome Trust Sanger Institute Cancer Genome Project, and they have made the collection of so-called cancer genes. And these are the genes. They basically make a census account of what are the genes that are commonly involved in the initiation process for the formation of cancer. And among the 387 genes that are listed in the cancer genes at the Wellcome Trust Sanger Institute, there are at least 44 genes that are involved in signaling pathways that regulate at least one of the triads of the transcription factors that control the glycolytic metabolism. Okay, and this is the list of genes, 44 genes. So I said a triad of transcription factors. So if we look at all the pathways that lead down to the enzymes, the regulation of the enzymes that involved in the glycolytic pathway, we can come up with 3 transcription factors that is very, very often, most frequently involved in many of the cancers. And these are the upregulation, increased activity of HIF1 alpha, and MYC. Okay. also a coordinated loss or decrease in the activity of the p53, either by mutation or loss of the genes or inactivation of the proteins. So these three transcription factors actually interact with each other through a complex network. And this is through the IPA. I think Dean the other day taught you all how to access the IPA continuity pathway analysis. Okay. So if you put in the IPA, okay, you put the HIF1 alpha MYC and p53 and ask them to expand, ask the software to expand to see what the connections are. You can see that there's a big network of connection and they mutually interact with each other and regulate each other. So in cancer, actually, the metabolic pathways are very often changed in the pathways that regulate the MYC and HIF1 and p53. So the pathways that are leading down to these three transcription factors, you can see that upstream, all these circled ones are known tumor suppressor genes that are mutated in many cancers, and there's oncogenes that are in the white, the circled in the white. So how do we know that? Because there are many genetic symptoms that are associated with the genomic mutation of these genes in the familial way. Okay? For example, these familial syndromes, Li-Fraumeni syndrome is the mutation of p53, the neurofibromatosis type 1 mutation of the NF1, neurofibromatosis type 2 is NF2, familial adenomatous polyposis is the ABC mutation, and then tuberous sclerosis type 1 is the PSC1, and then type 2, PSC2. Hertz-Jaeger syndrome is the mutation of the LKB1. Cowden syndrome, mutation of P10. Von Hippel-Lindau syndrome is the mutation of VHL. And multiple endocrineoplasm type 2, mutation of RET oncogene. So the cancer cell metabolism, p53, how is it regulating the cellular metabolism? So p53 mainly regulates the glycolytic metabolism. So you can see here that the complex, tetramer complex of p53 would actually mainly work by initiation of transcription. So this gene TIGA and the SCO2 are genes that regulated or induced by the p53. So if you have a mutation of p53, you lose the expression of these genes. So what is SCO2? SCO2 is critical for regulating the cytochrome C oxidase complex. It is a major site of oxygen utilization in the karyotic cell. And disruption of the SCO2 gene expression in cells with the wild type p53 would recapitulate the metabolic switch to glycolysis that is exhibited in the p53 deficient cells. So, and the TIGA is, SCO2 is called the gene name of gene synthesis of cytochrome C oxidase number 2. Okay, TIGA, T-I-G-A-R, it stands for TP53 induced glycolysis and apoptosis regulated. Okay, so this TIGA actually is an enzyme. So the TIGA has domains that is similar to the bisphosphatase domain of the 6-phosphofructose 2 kinase, or fructose 2,6 bisphosphatase. The abbreviation for these are PFK2 or FBPase2. So Tiger actually would catalyze to reverse a step in one of the steps in glycolysis. I'll point out to you later. So what it does is a Tiger expression would lower the fructose 2,6 bisphosphate level in the cell and result in the inhibition of the glycolysis and an overall decrease in the intracellular reactive oxygen species. And p53 also regulates the PGM. PGM is phosphoglycerate mutase. Okay. So, but this regulation is an inhibition. It is not by induction of gene expression. And this is through regulation of protein stability rather than a direct effect on the transcription. So by inhibiting the PGM or phosphoglycerate mutase, the p53 can suppress the glycolytic rate of the cancer cells. And also, through the IKK that regulates the NF-kappa B pathway, the p53 would also be able to inhibit the expression of glucose transporter type group. Now, MYC and HIF1, okay, p53 I already talked about. Okay. So there's about the SCO2 expression in Tiger. Okay, Tiger catalyzes this reverse reaction here. So it would decrease the concentration of the fructose 2,6 bisphosphate. Okay. So, and that would put a brake on this progress of this glycolytic pathway. So, on the other hand, HIF1 alpha and MYC are genes, transcription factors that regulate the expression of the majority of the enzymes involved in the glycolytic pathway. So you can see here that the red enzymes are the targets of both MYC and HIF. Okay, starting from glucose transporter GLUT1, and then hexokinase 2 all the way down here to the LDH type 8. These are regulated by both MYC and HIF. Now this is another version of the transcription targets of MYC and HIF involved in the metabolic pathway. So in this slide, I emphasize the difference between HIF and MYC. Okay? So the blue one are HIF targets only, and that is the carbonic anhydrase. Okay? And the PDK1. Well, actually, PDK1. PDK1 in this figure from this paper is probably before the other paper was published. That in fact, PDK1 was regulated by both MYC. The COX-4 is regulated by HIF only. And for MYC, okay, you can see that the fatty acid metabolism, nucleic metabolism, polyamine metabolism, amino acid metabolism. So, especially glutaminolysis and fatty acid synthesis, these are regulated by MYC only, not HIF only. And that is one of the major differences between the regulation of the metabolism between HIF1 and MYC. Now, the expression of the genes of all the enzymes involved in the pathway basically just set up the plumbing, set up the pipes. Okay? So exactly how fast does the liquid, does the metabolites flow through this system of plumbing is regulated by taps, regulated by faucets, okay, that you can regulate how much is flowing through the system. Okay, so what we call flux. Okay. So the flux of the metabolites is regulated in glycolysis, and hexokinase 2 is binds to the mitochondrial porins and then catalyzes the first step in glycolysis. Okay? And this is a strategic location. It binds to the mitochondrial porin, okay? And therefore it's away from the metabolite of the glucose 6-phosphate. Okay? So therefore it minimizes the inhibition by this metabolite. Glycolysis is stimulated by the fructose 1,6 bisphosphate, fructose 2,6 bisphosphate. So you can see that's why it's very important that when TIGA is catalyzing the pathway to decrease this, then it will decrease the stimulation by this metabolite. So that's why p53 through TIGA can put a brake on the metabolic pathway of glycolysis. So ATP also would inhibit the 6-phosphofructose 1 kinase, so PFK1 and 2. So this is a feedback inhibition by the energy source and energy currency. The tetramer dimer ratio of the PKM2, okay, they will control the pyruvate kinase activity. It is all regulated by different things, including ATP, amino acids synthesized from the glycolytic ingredients like serine, valine, fatty acid, and fructose 1,6 bisphosphate. So various metabolites along the pathway can also have a feedback inhibition or to regulate the activity of this enzyme. Due to the high activity of the mitochondrial bound hexokinase 2 in the presence of sufficient glucose, all phosphometabolites above the pyruvate kinase will accumulate until the levels of the fructose 1,6 bisphosphate and fructose 2,6 bisphosphate are high enough to activate the PFK1 and to shift the PKM2 into the active tetrameric form. So, because of these changes, okay, so as you measure the level of the metabolites along the pathway, okay, so what we call the metabolomic analysis. When you measure the level of these pathways, and you can see that these are different compared to the metabolites of the same pathway in the normal cells. So the PFK2 or FEPase2, okay, they regulate the level of the fructose 2,6 bisphosphate, which stimulates glycolysis by a potent positive allosteric effect on the PFK1 and inhibit gluconeogenesis by blocking the fructose 1,6 bisphosphate. So PKM2, okay, M2, pyruvate kinase M2 is actually a isoform. And it is a splice variant of the M1 isoform in the embryonic tissue. So M1 isoform is normally expressed in adult tissue, but cancer cells choose to express the M2 isoform. This expression of the PKM2 isoform is very important because in Dr. Canley's lab, there's 2 papers published in Nature, and they demonstrate the importance of this isoform. So if you knock down the PKM2 with the shRNA and then re-express the PKM1 in the cancer cells, this reverses the Wolverine effect. Okay? This decrease in lactate production and increase in oxygen consumption. So if you express the PKM1, it will correlate with a reduced ability to form tumors when the cells are transplanted or xenografted into nucleus. The PKM2 is necessary for aerobic glycolysis and provides a selective growth advantage to the tumor cells in vivo. The binding of the phosphotyrosine peptide to PKM2 would release the allosteric activator fructose 1,6 bisphosphate, leading to the inhibition of the PKM2. And the growth factor signaling would divert the glucose metabolites from energy production to the anabolic process. So AKT is also in the pathway under the tyrosine kinase receptor. And when you have activation of the tyrosine kinase receptors going through PI3 kinase, AKT would become activated. And AKT, in fact, is also a very important regulator of the cancer cell metabolism. Going through the GSK3 pathway, mTOR pathway, and NPM, it will regulate different parts. Actually, they will regulate all three of the transcription factors that form the triad that regulate the glycolytic pathway. And through MAKE, there's also regulation of the glutaminolysis. So the HIF1 and MAKE regulate the transcription of most of the enzymes involved in glycolysis. But AKT also can phosphorylate the glucose transporter and activate the increased glucose transport into the cells. And another important aspect is the phosphorylation of CPT1 that regulate the beta oxidation of fatty acid. But AKT also would regulate hexokinase 2 for the glycolysis, and AKT also regulate the fatty acid synthesis as well. So overall, the big picture is that you started out with the genetic mutations, and then you lead to the imbalance in the genetic makeup of the cells. Okay, and the cells will actually have, because of the increase in genomic instability, then the balance, regulation of the genes will become lost. Okay, when it's imbalanced, basically the overall picture is that if you follow these changes, the signaling pathways would almost always lead you to these three transcription factors: the MYC and the HIF1 and the p53. There's a coordinated chain. Okay, increase in the activity of HIF1 and increase in the activity of MYC, but decrease in the activity of p53. And with the coordinated changes of these three things, okay, many things will follow. You have the changes in the metabolites, mitochondrial changes, you have changes in the energetics and the utilization of energy, and you have changes in the synthesis of all the building blocks for the cancer cells, and this is mediated through glycolysis, fatty acid synthesis, and glutaminolysis, and that would give the cells growth and survival advantages. Now next we talk about glutaminolysis. So anybody have done tissue culture yet? So in the tissue culture lab, at the stem cell lab, you guys have the experience of culturing the cancer cells in the food, okay, and in the incubator. Okay. Do you all know what is in the culture media? Okay. Now the culture media, because glutamine is not stable, so when you do cell culture, you bought the culture media from the supplier. Some of the glutamine might have degraded already. So before you use the culture media, you add or resupply or replenish the glutamine in the culture media. Okay, and very often you would add serum to the culture media. Now, the glutamine is actually a very important energy source for the cancer cells. And also, you might have pay attention to the composition of the tissue culture media. Glucose is also very important. And some cells grow very well, don't need a whole lot of glucose, and grow well with normal concentrations, or roughly about 1,000 milligrams per liter. But many of the culture media, or some of the cells that are more fastidious, they love glucose, and many of the culture media would actually have high per liter. Okay. So glucose would be able to supply the cells through glycolysis, okay? And the glutamine is going to supply the cells through glutaminolysis. And glutaminolysis is here, this is the pathway that is connecting the glutamine to the glycolytic pathway. So glutamine is taken up into the cells and is metabolized to glutamate. Glutamate is metabolized to alpha-ketoglutarate and enters the TCA cycle. Right? So in the TCA cycle, it would be metabolized in this direction to malate, and then be metabolized to pyruvate. And these are connected to the lactate. Okay. So when we do experiments, okay, so as you can see here, lactate can come from gluten. Okay. So when we do experiments and measure the glycolysis in the experiment, okay, if we are only measuring lactate production, okay, so we have to be careful